Circulating tumor cells, also known as CTCs, are tumor cells that have metastasized from the tumor and entered the bloodstream. Early detection of these cells could lead to an early cancer diagnosis with an increased chance of success during treatment. This even means that your yearly physical could reveal whether you have cancer or not. Until now, many CTC detection procedures were very complicated and expensive, and electrochemical techniques weren't sensitive enough to detect low concentrations of cancer cells. This video will explain electrochemical ELISA, a revolutionary technology that uses electrochemistry to identify even the lowest concentration of cancer cells within a blood sample. ELISA is a technology that relies on antigen-antibody reaction. It stands for Enzyme-Linked Immunosorbent Assay. This is a procedure that involves a couple of steps. At first, the blood sample is run through a small 4mm by 6mm chip. This chip is covered with X-shaped posts. The unique shape of the post allows the absorption of cells to occur. Keep in mind that both healthy cells and cancer cells are absorbed. This is a picture of a cancer cell caught by an X-shaped post. Since we are able to see it, it means that it has been illuminated using the following steps. Most cancer cells possess a protein called cytokeratin 18. It's usually called a cancer marker. This protein is used as a way to differentiate cancer cells and healthy cells, as only cancer cells will bind to the cancer antibody. The plate is then put through an antibody solution. The anti-CK18 will only stick to cells that possess the cancer marker cytokeratin 18. So how do we ensure that the antibody-antigen complex doesn't get washed away with the cancer cell? Well, the antibodies are tagged with magnetic nanoparticles before the solution is put onto the chip. The magnet ensures that the antibody stays in place after the enzyme solution is put through the chip. The enzyme, ATP, turns p-aminophenol phosphate into p-aminophenol. The hydroxyl group makes the molecule electrochemically sensitive, allowing it to light up and illuminate when it's run through with -30 millivolts. The intensity of the light will vary based on the amount of cancer cells that have been tagged. This technology is extremely sensitive, detecting as low as 2 cells per milliliter of blood. Overall, these researchers were able to develop sensitive, cost-effective, cancer-detecting technology. Since it can be modified to employ different antibodies, this system can readily be adapted to different cancer cell types. This concludes this video. Please make sure to check out the original study in the description box below.